Protein can be divided into three types of classification, which are classification based on shape and structure, classification based on products of hydrolysis, and classification based on biological function. Classification based on shape and structure are further categorized into two broad classes: globular protein, fibrous protein. Based on products of hydrolysis can be classified into three categories: simple proteins, conjugated proteins, derived proteins. Classification based on biological function are as follows. Shape and structure. Globular proteins, also called spheroproteins or corpuscular proteins, contain compactly folded coils of polypeptide chain. It can soluble in water. Examples of globular protein are albumin, globulin, and lysozyme. Fibrous proteins have an elongated shape and look like fibers or thread and have axial ratio of more than 10. Insoluble in water. Can be divided into following types: alpha-keratins, beta-keratins, collagen, and elastins. Alpha-keratins can be found in protein fibers of hair, skin, and wool, constituted of a number of alpha helices that are twisted together like the strands of a rope. Examples of alpha-keratins is fibrin, which involved in coagulation of blood. Beta-keratins can be found in hard tissues such as nails, fish scales. Acquire a beta-pleated sheet structure. Examples: silk fibroin, which can be obtained from the cocoons of silk moth. Collagen. Collagen is main components of bone, teeth, connective tissues of tendons and ligament. Polypeptide chain of collagen acquires an elongated left-handed alpha helical and three such helices spiral around each other like strands of rope. Elastins closely related to collagen and constitute ligaments in the walls of blood vessels. Simple proteins, only composed of amino acids, do not contain any non-protein part. Most of simple proteins are globular type. Example of sample protein as follows: albumins, can soluble in water. Some of animal albumin are serum albumin, ovalbumin, and lactoalbumin, while for plant albumin are leucosin, legumelin, and ricin. Globulins, ellipsoidal in shape, insoluble in water but dissolve in aqueous solutions. Some of animal globulins are serum globulin, myosin, and lactoglobulin, while for plant globulin is edestin. Histones is nucleoprotein, proteins found in the nucleus of the cell. Can dissolve in water and dilute aqueous solution but insoluble in dilute ammonia. Not easily thermocoagulable. Protamins can dissolve in water and dilute aqueous solution in ammonia. Not easily thermocoagulable. Rich in basic amino acid, arginine, and behave more like a polypeptide. Prolamins are insoluble in either water or alcohol. Not thermocoagulable. Rich in glutamic acid and proline. Examples are gliadin, hort9, and zein. Glutellins, insoluble in either water or alcohol, can be thermocoagulated. Examples are glutenin, glutellin, and oryzanin. Scleroproteins are fibrous protein of animal origin, insoluble in water and dilute aqueous solution of acids. Conjugated or complex proteins. Conjugated proteins are proteins that form complexes with some non-protein component. In conjugated protein, protein part is called apoprotein, while non-protein part is called prosthetic group. The entire molecule is called haloprotein. Generally globular in nature. Major classes in this type as following: phosphoproteins, contain phosphoric acid as prosthetic group. Do not include phosphate-containing substances like nucleic acids. Phosphoric acid group is connected through ester linkage to a hydroxy group on the polypeptide chain. Examples are casein, milk protein, and ovovitelin, obtained from egg yolk. Chromoproteins, proteins containing colored pigments like flavins, carotenoids, porphyrins as their prosthetic groups. Examples of chromoprotein are hemoglobin, myoglobin, and cytochrome. For enzymes such as catalase, peroxidase, and cytochrome oxidase. Chloroplastins, plant protein containing chlorophyll, also example of chromoprotein. Glycoproteins, contain a carbohydrate component as the prosthetic group, constitute from less than 4% to more than 80%. Oligosaccharide chains are joined with the polypeptide through O-glycosidic linkage with serine or threonine or by N-glycosidic linkage with asparagine. Examples are immunoglobulins, thyrotrophin or thyroid-stimulating hormone, TSH, follicle-stimulating hormone, FSH. mucopolysaccharides. Contain a small protein component in a carbohydrate structure. Found in the supportive and connective tissues in animals. Examples like hyaluronic, heparin, and chondroitin sulfate. Metalloproteins. Protein molecule is linked with metal ions. Examples: siderofillin, strong affinity for iron and involved in iron transport; ceruloplasmin, affinity for copper ions; nucleoproteins. Contain nucleic acids and basic proteins like protamines and histones that bound through electrostatic interactions. Important constituents of chromosomes, ribosomes, and some viruses. Lipoproteins. Includes proteins complexed with lipids and have a variable composition. Important constituents of blood plasma, cellular membranes, milk, and egg yolk. Lipoprotein of lowest density kilomicrons, 2% of protein. Very low density lipoprotein, VLDL, 9% of protein. Low density lipoprotein, LDL, 21% of protein. High density lipoproteins, HDL, 33% of protein. Increase of LDL can increase the risk of arteriosclerosis. Derived proteins. Degradation products derived on subjecting the native proteins to different physical or chemical agents like heat, acid, alkali, or enzyme. Primary or denatured derived proteins. Obtained by the action of water, acids, alkalis, heat, radiation on the native protein. Denature the protein without hydrolyzing the peptide backbone and do not change the molecular mass. Only weaker forces like hydrogen bonding, hydrophobic interactions, salt linkage maintain native structure of protein are disrupted and only solubility, precipitation are altered. Secondary derived proteins. Obtained by the processes that cause progressive hydrolysis of the native protein. Products obtained in the initial stages of hydrolysis, with dilute acids, alkalis, and enzymes. Biological function. Enzymes. Also called as biochemical catalysts. Involved in catalyzing biological reactions. Example: dehydrogenase, catalyzing dehydrogenation; kinases, involved in transfer of phosphate group. Storage proteins. Storing important species in the living cell. Example: myoglobin, oxygen storage in skeletal muscle; ferritin, involved in the storage of iron in the liver. Regulatory proteins. Control many aspects of cell function. Example: insulin and glucagon, regulate body functions. Structural proteins. Provide mechanical support to large animals and their outer covering. Example: collagen, provides mechanical strength to our teeth, bones and tendons. Protective, or defense, proteins. Defend the body against infection. Example: immunoglobulins, bind to an antigen and cause its destruction, venoms and toxins. Transport proteins. Carrying materials from one place to another in the body. Example: hemoglobin, transport oxygen; transferrin, transporting ferric ions in the plasma. Contractile and mobile proteins engaged in all forms of movement. Example: actin and myosin, responsible for heart movement and muscle movement; tubulin, sperm movement.